In this video, we will cover an general view or an overview of the glycosylation of viral protein from mosquito-borne borne Flavivirus. In the previous video, we have covered just a part of the Flavivirus. However, in this video, we will cover different aspect of the virus infection and how this glycosylation plays a role. So, as we have discussed in our previous video, that glycosylation plays a role in the viral attachment and internalization, as it is shown here. And in the previous video, we have covered the two sites of glycosylation, that is at 67, asparagine at 67 and asparagine at 154. And uh, these things are conserved in different kind of flavivirus, that is dengue virus, Zika virus, WNV virus, whereas this asparagine 67 is found to be conserved in dengue virus itself. So this glycosylation at this sites or N-linked glycosylation helps in this flavivirus attachment and entry, and which finally leads helps in the internalization. After that, the because of the pH change in the endosome, there is a genome get exposed to the cytosols, and then it will proceed in two different pathways. First one is for replication, and another one is for translation. And this translation process goes through the endoplasmic reticulum, as most of the N-linked glycosylation starts from within the ER lumen. After that, the packaging takes place, and this translation of this various genome has been shown on the left hand side. After, from the endoplasmic reticulum, in the ERIC, that is endoplasmic reticulum intermediate Golgi compartment, there the assembly of the various structural protein occurs. And one more thing we need to know, that the flavivirus, the premembrane part in terms of structural protein, the premembrane part and the premembrane protein and the envelope protein has glycosylation, whereas in case of non-structural part, NS1 protein has the glycosylation. These things are being reported. So when from the endoplasmic reticulum and from different and from other translation ways, the structural and non-structural part get assembled in the ERIC compartment. After that, they will move in the Golgi for further post-translation modification of N-linked glycosylation and the initiation of, or if there is any O-linked glycosylation, that will also occur within the Golgi. After that, what happens is that the virus will release in two different ways. First, that it will remain in the cytosol and cause cytotoxic apoptosis. That is, it will try to kill the cells or use the cell machinery for the replication of its own genetic material. Another thing, that when the virus is released to the environment, this glycosylation of the virus helps itself in the survival within the mosquito, and also glycosylation of this virus prevents itself from the uh, immune response of the mosquito. Or in other way, we can say glycosylation helps in the evasion from the immune system of the mosquito. After that, the virus can be transferred from one person to another person with the help of this mosquito. Replication and infection occurs within the mosquito. So this is one of the examples. This is one of the example how this dengue virus spread from one person to another through mosquito. And another thing the glycosylation does is uh, it affects the half-life period of the virus within the mammalian cells, because glycosylation provides the shielding, or other term it will provide glycan shielding, so that the immune system of the host will not be able to recognize the foreign epitopes present on the virus. virus protein as they are being shielded by the glycans. And glycosylation profiles are usually same or it replicates close to the host glycosylation profile. So it is very difficult for the immune system to recognize different glycosylation profile also. And these are being shown in this pathogenicity, where the half-life period will also get affected because of the glycosylation of the viruses in the mammalian cells, and thereby it will also affect in the human beings, as human beings are the part of mammalian cells. So these are some of the brief overview how this glycosylation of the viral proteins from mosquito-borne flavivirus helps in the viral infection at different stages. If you need to understand this the role of glycosylation in detail in the viral infection, then you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.